We had a problem with our car recently where it sort of wouldn't start once every 3 weeks, the kind of problem you hate. It went in for some maintenance yesterday. They couldn't find the reason. So we left it there overnight because my husband's glasses were blown off by the rainstorm. And went in today when I finally had time. And since it had been out in the lot under the rain all night, now it misbehaved. And now they could figure out the problem was. So clouds do sometimes have silver linings. And I hope you didn't get so wet that your glasses were blown off. We did find them by going back. Do people have any questions they'd like to ask about DNA replication before we go on to DNA repair? All right. Then let's move ahead. Sure, I can do that. That's why I have the sound checks. Is that a little better? Can you hear me better now? Is that better up there? Okay. Lynn was talking about the standard process of replication, sort of what needs to happen on a regular basis. And obviously, that's an important thing because we have to have ongoing renewal to keep our bodies going and replication to have growth during development and so on. But the environment is a noisy and sometimes nasty place. And sometimes DNA is not perfect. It's not perfectly replicated, or it's damaged after it's replicated. And it becomes necessary to somehow or another to fix the DNA. And what we're going to be looking at in this chapter is a couple of aspects of DNA repair. On the one hand, cancer can be understood as a disease that has at its root a failure of DNA repair, although there's a lot that goes into that, and we'll unpack that a bit. And secondly, that the cell and the body have immense resources for doing DNA repair that are going on all the time and constantly in surveillance to keep us intact and healthy and alive. So let's talk a little bit about cancer, because it's a topic of general interest, and it's very likely to have affected most of you either already or in the future. Just offhand, how many of you either have had cancer or have a family or a friend, family member or friend who's had cancer? Could you raise your hands? This is not a trivial topic. Cancer is not a single disease. If you compare the different kinds of cancer, it can be anything from a slow-growing massive tumor that takes decades to get to the point of causing damage to a rapidly growing fulminant form that can, from start to finish, can be completed in 2 to 3 months. So it attacks different organs, it attacks at different rates, it has different manifestations. Even for all that variety, there is still a sense in which all cancers are somehow or other problems in the DNA. Either there were errors made in DNA replication, either there's incomplete replication or noisy replication where odd numbers of things are created, or there's been damage to the DNA itself. And we'll be exploring some of those as we go along. Let me get my cursor here. Okay. To give you a sense of just how good DNA repair can be at its optimum, I'd like to introduce you to an organism called Deinococcus radiodurans. And Deinococcus means terrible bacterium. And radiodurans, as you might guess, means tough against radioactivity. It is the most radio-resistant or resistant to radioactivity organism that we know. To give you a comparison, you can kill a human being with 10 grays, which is 1 unit of radiation. E. coli, the bacteria, are a bit tougher. It takes 50 grays. Radiodurans can be exposed to 5,000 or 500 times what it takes to kill us and still be happy. This ability to repair its DNA confers on Deinococcus radiodurans amongst the most extreme capacities to live in different environments. So it can stand up to radiation, it can stand up to damage by acid, by cold, it can be dried in a vacuum and recover, because the machinery that repairs the damage from radiation can repair damage from a wide variety of sources. These are just pictures of them. This is a picture at the light microscopic level, and you can see the kind of tetrads that sort of form. That's a typical coccus presentation. And in the electron micrograph just underneath it, you can see a very substantial, dense area of DNA, which is part of the trick that Deinococcus uses to survive these extraordinary attacks. It was found by accident during the period when they were trying out radiation as a way of sterilizing food. Sort of sidelight, they wanted to find a way of talking about that sounded a little more inviting, and they came up with the phrase cold pasteurization, which I think is an interesting triumph of Madison Avenue. And they came across these cans that were contaminated despite what should have been killing doses of radiation. Because of its intense ability to repair its DNA, it is being considered for its potential in bioremediation of radioactive waste sites. And part of its trick is that it has multiple copies of its genome. A typical E. coli might have 1 or 2 copies of its genome. The Deinococcus has like a lifesaver ring of multiple copies laid down side by side. And you can see here staining for the DNA. This is an extraordinarily dense DNA core for a bacterium. And here again you can see them in a little more detail than before. And to give you a sense of just how good the repair is, this is a picture of the DNA of Deinococcus immediately after a high dose of radiation. The DNA is in little bits and pieces. And within 36 hours, the organism has been able to restore the continuity of its DNA. I find this absolutely stunning, that you can go from bits and pieces of DNA that look like confetti and come back up with an intact, functioning DNA. So what is cancer? I've mentioned that the different tissues and the different forms of cancers can have different presentations in morphology, in virulence, in speed of growth, and so forth. The fundamental error in all the cases really comes to 2 aspects. One is of uncontrolled growth. Not necessarily fast growth, but uncontrolled growth. Slow growth that is uncontrolled and continues past the time it should stop is potentially as much of a problem as fast growth. So the key is everything in our bodies normally has some cap to its growth, and if that control is gone, the potential is there then for cancer. The second aspect of cancer is a failure to die. Lynn, I believe, mentioned the idea of apoptosis, or maybe I'm thinking of Thursday's seminar. Apoptosis is a program by which... Do you think I should answer it? Hello? Your telephone is currently in EMR, Medical Education and Research Facility. So we'll see if that works. What else can I do? So apoptosis is an inherent program for cell death, and that's a good thing. If you did not have programmed apoptosis, your hands would look like a duck, because normally during development there's cells in between, and in the course of embryology, those cells in between are programmed to die, and they go away, and now you have separate fingers. So apoptosis is a very powerful tool that the body has to use both during development and as a part of the ongoing surveillance of our bodies to make sure that there's nothing going on that shouldn't be there. And so some of the cues for apoptosis are things that are triggering the body to recognize that something alien or wrong or whatever is going on. These are always linked in one way or another to alterations in the DNA, either alterations in expression or alterations in the sequence of the DNA. And we'll be looking at what some of those alterations specifically are in a second. To give you a sense of the dimensions of it, 16,000 cases, new cases a year in Iowa alone. This is an example of one presentation of cancer. This is a liver, section through the whole liver. And you can see these masses here, each one of which is a tumor. I would guess probably metastatic from something else, but I'm not a pathologist. Oh, these things are not Nice. They moved around on me. Oh, well. So some of the things that can lead to cancer. There's the classic cartoon of the kid with the bad report card talking to his dad, and he says to his dad, "Okay, Dad, which is it, genes or environment?" No, it's not his fault. Some of our cancers come from our genes. Some of our cancers come from our environment. Of course, we sometimes pick our environment. And some of the kinds of things that can do this include, you're all familiar with the fact that smoking or sunlight can do this. There are some viruses like hepatitis. Hepatitis actually is one of the ones, but also I was thinking of papillomaviruses, asbestos, nitrosamines. These share in common the fact that they are capable of damaging genes, that they can cause damage to the DNA. Another kind of predisposition is a genetic predisposition which should have aligned with the DNA repair genes. That is, if you have deficits in the DNA repair, that can also cause cancer by failing to restore things after they've been damaged. And although cancer is traceable to genes, it's traceable to genes doing either of two things that seem on the face of it to be contradictory, genes that are either overly active or inactive. And we're going to see which things contributes there. To introduce you to some of the terms that we use when we think about DNA cancer as a genetic disease, we call something a proto-oncogene if it is a normal gene in a gene that the cell routinely does use to control growth and or differentiation. We call it a proto-oncogene because it isn't itself a cancer gene. It is simply a precursor that could become one. And if it is altered in a way that now it can contribute to cancer, we call that an oncogene, a cancer gene, because it's an activated version of this proto-oncogene. So this is a gene which, when it's doing something, can push things towards cancer. The other big class of things are tumor suppressor genes. And tumor suppressor genes are kind of named for the way they were found. They were found because in their absence, tumors tend to occur. If you lose them, you're going to develop cancers. Their real role in normal development is a little more subtle than that, but certainly one of the things that we benefit from is that they suppress tumors. So if we want to compare the genetic activity in a normal cell to the genetic activity in a cancer cell, it's the balance of these two contrary forces that leads to either a normal state or a disrupted one. So in a normal cell, you would have a balance of activity such that the proto-oncogenes, if they're at X level, the tumor suppressor genes are at a higher level than that. We'll just take this as the ground state for it. You can go from a normal cell to a cancer cell by changing either part of that equation. So transformation, turning into a cancer cell, can happen because you get more expression of the proto-oncogenes or because you get less expression of the tumor suppressor genes. Obviously, you could have both happen, in which case you might have one of those really nasty ones. But it's the balance of these two kinds of activities that keeps things either in the normal state or can get disrupted to turn into cancer. Some more sort of vocabulary for cancer. Carcinogenesis is the overall process of the development of cancer and its ongoing changes in development. Transformation points to the moment in which you say this is no longer a normal cell, it really is now a cancer cell. And tumor simply means a mass, a large amount of cells. And this is a sort of a schematic diagram of carcinogenesis, but it highlights a number of important steps in it. So here we have our normal cell to begin with that has a good balance of the proto-oncogenes and tumor suppressor genes. Something happens, a carcinogen of some kind, carcinogen thing that causes cancer. Something happens that causes a genetic change of some kind, and we're going to see some of the kinds that might be relevant. We call that initial change, that first change that alters things and starts them down that path, initiation. It has to begin somewhere. But that cell can continue with that genetic change for a very long time. It may go through multiple rounds of cell division and show no signs that it's been exposed to this initiating event. The next step that happens is sometimes called promotion. Something in the environment that triggers that genetic change to be expressed in a way that now leads to multiplication of the cells in an uncontrolled way. More changes are going to be needed genetically to keep going down that pathway, to continue through carcinogenesis. And it's around this point that we speak about transformation, when cells are not just multiplying in an uncontrolled way, but they've changed genetically so now they can be considered malignant. A malignant cell is now going to be an immortal cell. It's going to be one that does not respond well to its environment. It does not play well with others, and so on. But it's not the end of the line. Even more bad things can yet happen beyond this. And if other genetic changes continue to happen, we can have progression until we have a tumor, a mass that's detectable, a mass that's generated its own blood supply, a mass that's actually able to escape and create metastases. So it's a multi-step process, and I think this is something that was not fully appreciated until fairly recently. Certainly it wasn't fully appreciated that there were multiple genetic steps that had to happen. It's not like normal cell, one change, cancer cell. It's, you know, checklist. How far down the checklist have you gone? So this is another way of drawing that picture, showing you that if you start off with a cell at birth, multiple mutations can happen in this pre-tumor stage of progression, but that you accumulate some number of them before you actually have a tumor that's going to continue to progress and get possibly worse still and possibly metastasize. One key thing to keep in mind is that there are these multiple mutations, but they are not necessarily going to happen in a sequence. This one might happen first in this cell. That one might happen first in a different cell. The sequence of the mutations is not relevant. They can happen through DNA damage. There could be substitutions of bases. You can actually lose a base altogether. You could have a modification on it so that it's misread. In some cases, you have genes that you got from birth that were, wrong from birth. It's quite common for there to be several pathways that have to be affected. That's part of what's being captured here in these multiple mutations, that a single pathway is not enough to make a cancer cell. It's going to be multiple. And that's reflected in the fact that it takes multiple hits to have that to happen. So what are some of the processes that have to be targeted in a normal cell to get these multiple hits that will create a cancer cell? Well, there are several genes involved with cell division. Mitogens are compounds or hormones or signals that signal for growth. And so one kind of alteration that can happen is that you have a growth signaling pathway that's permanently on. It no longer needs to get a signal to go. It's going to be set to go in any case, kind of like a traffic light stuck on the green. Another area that is a target that is always involved is apoptosis. Since apoptosis is part of the ongoing surveillance problem, if you develop a resistance to the signals that say time to die now, then cells will not die at the times that they should, and they'll be around to accumulate more errors and to go on growing. Lynn talked a little bit about telomeres, that in normal cells we start out with a certain length of telomere, and that those tend to shorten over lifetime, but that in cancer cells the gene for telomerase may be turned on, and turned on inappropriately or at elevated levels, so that the telomeres, instead of getting shorter, either stay the same length or get longer, And now, instead of the cell dying at 50 divisions, which is a typical age for a human cell, it can go on through multiple divisions and continue to grow and accumulate. Another aspect of normal cell behavior is how cells relate to each other and to the thing that they grow on. And we call these two processes contact inhibition. That is, normal cells don't like to be too crowded. They'll grow up until they meet each other, and then that's fine. Cells that are going down that pathway towards cancer at some point lose that inhibition so they can pile up on each other and continue to divide even though it's getting too crowded. Another aspect of the normal behavior of cells is anchorage dependence. Most cells have something they like to sit on and be attached to go through cell division. An aspect of that progression is to lose that anchorage dependence so that cells now can be promiscuous about when and where they're going to go through division. And so changing these two properties means that you can get crowding of cells, a denser population than you ought to normally, and you can have cells that can run away and grow in other places. And these are properties that are visible. So here is a scanning electron micrograph of normal cells. These are embryonic fibroblasts growing on a culture dish. They're spread out. They're playing nicely with each other and not getting too tight. And here you can see cancer cells in which they're very crowded together. They're not very spread. They're starting to pile up on top of each other. A little sidelight on the importance of realizing that it's cells that become cancer and not genes that become cancer. There's an investigator at MIT who was in the forefront of this idea of oncogenes. And he had been a student of my mentor at MIT and wanted some pictures for Science when he wrote a review article of some normal and some transformed cells. So my mentor sent him some. And this guy called my mentor up and said, "Oh, they're beautiful pictures. Can you tell me which one is normal and which one is transformed?" It's like, you should be able to tell by looking. I mean, look, normal, transformed. If you know the genes and you can't tell those two kinds of cells apart, you're missing part of the story. There's another important part of the story, which is getting enough to grow on. All cells that are growing need a certain supply of nutrients. And in the normal structure of the body, you have whatever blood supply you need to maintain that normal level. That's why the liver has a lot of blood supply and our cartilage has very little. For the growth of a tumor beyond a certain size, it has to acquire those resources. It needs a blood supply to grow beyond a certain limit. And we call that process angiogenesis, which is a Greek way of saying growing blood vessels. And so here you can see a little diagram of this, that you have a small tumor here that's releasing growth signals here and signaling to these blood vessels to come in and start supplying this little tumor with a blood supply. And here you have a larger one that has succeeded in recruiting a blood supply and now is getting the nutrients to continue to grow. If there's no blood supply, that tumor will not grow any bigger than a couple of millimeters. And there's a considerable research interest in drugs that would inhibit this angiogenesis as a way of targeting cancer growth. So far, only works in mice, but the day may come. So knowing this, knowing that cancer cells have to have a certain number of hits of certain kinds, provides us with several targets for drugs that are going to treat cancers. And so we want to treat features that are going to be common amongst, that's not the size I thought it'd be, certain kinds of features. One obvious one would be to suppress cell proliferation. If cells are not going to divide as fast or don't divide at all, you would assume that this would help keep them down. This particular compound here, which you can, I think, make out, is called Gleevec, is something that targets one of these kinds of signals. There's a chromosomal rearrangement called BCR-ABL for the two different genes that have been smushed together in a chromosomal rearrangement. And so there's a hybrid enzyme that is involved in signaling continued cell growth. And it's this hybrid enzyme that's behind a chronic myelogenous leukemia, the kind of leukemia that's kind of going on all the time and kind of wearing away at you. And because Gleevec can bind to that hybrid tyrosine kinase, it can both inhibit the cell proliferation and permit apoptosis to proceed. Gleevec is fairly specific by comparison to some things. Other things simply target cell division. Obviously, cell division is something that's happening in our bodies in a lot of places, like our guts and our hair, and that's one of the reasons why these less specific forms tend to have side effects like GI disturbances and losing your hair. Radiation, again. Radiation tends to most affect rapidly growing cells, and so that's one of the reasons for picking on radiation as a way to try to target cancer cells. But again, it's not as specific as some things are. It's not nearly as romantic to mention, but I refuse to not say it, which is that prevention is something to also keep thinking about. The kinds of things that are good at preventing cancers make sense in terms of this multi-hit model. Smoking is a process that's going to, on an ongoing basis, expose the body to multiple mutagens and carcinogens and promoters. Sunlight causes damage through UV irradiation. Healthy eating works well because some of the compounds that are in, quote, good foods, bright, colorful foods, are things that are antioxidants or that can stimulate the repair machinery, keeping down exposure to chemicals that aren't nice to us. There probably are some microorganisms involved in cancer. That's one reason, for instance, for the HPV vaccine. There are probably at least a few others that can be figured out so we can avoid those. Half, probably half of the cancer deaths could be avoided through prevention. And it's much more pleasant than going through chemotherapy. Much more pleasant. Give me a good tomato any day over Gleevec. We're beginning to get new tools for understanding and for analyzing cancers. Some of you may have heard of things called gene chips or microarrays. These are things that allow us to look at hundreds, thousands of genes and whether they are expressed in higher or lower levels in a particular cell. And so this is a picture of an array comparing a particular group of cancers against normal cells, showing that there's some things that are turned down relative to normal, other things that are turned up relative to normal. And this allows us to distinguish between things that might look similar at the morphological level. One help is the idea that it can provide help with diagnosis to distinguish subclasses that aren't necessarily obvious morphologically. The hope is that it'll someday allow us to do more specific treatments. I would say that's still in the hope level, but without knowledge, it's hard to develop specific targets. So, how do genetic alterations arise? We've heard from Lynn how you get things right. Now we're going to talk about getting things wrong. And there are two major classes. DNA damage that damages existing DNA and mutations in which alterations occur. I don't know if that's the right way to phrase that. In some cases, there are errors made during copying. In other cases, the DNA is correct but gets damaged, so it's no longer correct. And we're going to be looking at the fact that the cell, in fact, can recognize errors and can do something about them. And this all takes good enzymes. Some of the environmental sources of DNA damage, there are chemical mutagens. So, for example, the nitrosamines in cigarettes. Ionizing radiation. You can hear in the name ionizing, things that get little ions in the middle of your cells, probably not good for you. UV light has sort of an interesting kind of damage. So if in your DNA you've got two thymines side by side on the backbone, they're lying around like this, UV light can actually generate new bonds like that that pairs those thymines. This is now called a thymine dimer. You can see here that the backbone is a little kinked at that spot because those two thymines are now constrained in ways. That's not a good place for replication or transcription, so you'd want to be able to cope with that. This is looking at some of those chemical mutagens in a little bit more detail. Here is one of them. This is benzopyrene. You can look at that, and that kind of looks sort of like a DNA base pair, doesn't it? So you can imagine that that can contribute to putting it in place for bad things to happen. It can actually become added on to DNA bases chemically. Here's aflatoxin, another rather nasty one. This is a product from molds that can cause DNA damage. And our bodies process this metabolically until it becomes reactive and, again, can add on to the DNA chemically. Just the fact of being alive can be bad for your DNA. We are aerobic organisms. We have to have oxygen to live. And unfortunately, in the process of using oxygen, it's possible for oxygen radicals to form. So this is O2 minus with one electron there. Oxygen radicals are fairly reactive species. And if they add to guanine, you can have 8-oxoguanine, which Lynn also mentioned, I believe. Right? You didn't mention that? Okay. Anyway, here's 8-oxoguanine. And you can see here the oxygen added in there. Problem with 8-oxoguanine is that it now has potential for hydrogen bonding in new ways, and it can pair with C or with A. So this can convert a GC pair into an AT pair. So that would create what we call a point mutation, where a single base is altered, which might not make any difference. As we looked at the genetic code earlier, it's a pretty robust code, and one single difference isn't always a problem. On the other hand, it can be enough to completely kill a protein, depending on what the change is. There's even some damage that just happens because life is like that. Things that just spontaneously happen because the world's a noisy place. One is that the ribose base, or the deoxyribose base, can lose its base. The deoxyribose sugar can lose its base, so that the bond between the base and the sugar breaks and leaves just an alcohol there. This we call an abasic site. You can have a cytosine that is oxidatively loses its amine group and now looks like this, so it gets turned into a uracil. And you may remember that uracil base pairs the way thymine does. So this is would change the coding at that spot. Uracil is detected in DNA at a significant rate. And so this is something that can happen. So what would happen if all of these different potential kinds of nasty stuff happened and were left unrepaired? Well, at the smallest level, if you have a point mutation or a small insertion or a small deletion of a gene that's important, you can have that encoded protein be altered in its activity. Maybe it's lower activity, maybe it's not regulated anymore, maybe it's too active. If the damage is more serious, you can actually stop replication and transcription so that the whole works gets gummed up. And in that case, you may trigger, if you're lucky, apoptosis and the cell will die. Or if you've escaped from that regular control already, maybe these alterations are ones that lead to transformation. And so these are obviously not good things. And cells have a lot of machinery to help avoid these bad outcomes. Okay. Now, there are some simple repair enzymes where a single enzyme can do something all on its own. You may remember I mentioned the thymine dimers here. In bacteria, there's an enzyme called DNA photolyase, which is kind of cute because the thymine dimer formed from UV light. And UV light will then activate the photolyase to repair that dimer back to a plain TT pair. So sort of, you know, your poison is also your cure, so to speak. Another little interesting enzyme is methyltransferase in mammals. Here is O-methylguanine. You can see a methyl group there that ought not to have been. And methyltransferase can remove that methyl and transfer it to an amino acid and restore you to a normal-looking guanine. There are other repair mechanisms that aren't quite that simple, where there's more that needs to happen before it's all done. And we're going to talk about several of them: mismatch repair, base excision repair, and nucleotide excision repair. Okay. So in mismatch repair, the gene system that is involved in this is called MUT-S, and you can guess that MUT-S gets its name from the fact that this is a gene that was discovered in the context of mutations. And MUT-S is monitoring new DNA. So it's going along a little bit behind the DNA polymerase and just kind of checking things out. And if it finds a mismatch, it will then recruit the nuclease that's going to cleave the DNA on the strand that's got the mismatch. Now you might ask, how do you know which is the strand that's the wrong strand? Well, we've talked about methylation. In the sort of resting state, if you like, or in between rounds of synthesis, both strands of DNA will be methylated appropriately. The newly made DNA hasn't had a chance to get modified yet. So if you find a mismatch, the strand that's not methylated is the one that's the mistake. And that's why it targets that strand. And it'll unwind the helix a little bit there, take out that error in the DNA, and have it replaced. Another machinery for fixing things is the base excision repair. I've mentioned that there are a number of ways in which bases can get damaged one way or another. And base excision repair can detect regions where there's damage. And that base is removed through a DNA glycosylase that can cut the base off of its sugar. And now we have an abasic site that we've generated. We created this abasic site intentionally. At this point, an endonuclease can cut the backbone, and a DNA polymerase and ligase can come in, restore a base pair there, and seal back up the backbone. So what are some of the glycosylases that can recognize these things? I mentioned that 8-oxoguanine is not a good thing. There's an 8-oxoguanine glycosylase that will bind to 8-oxoguanines as places that need to undergo repair. And if you look at the pictures, you can get a sense of this, of why that would be so. So it's comparing here the way in which the normal and the damaged G's interact with the helix, and that the damaged G gets flipped out of the helix, and in doing so, the glycosylase can hold on to it and cut that damaged base off. Another glycosylase recognizes uracil in DNA. Again, uracil is not supposed to be in DNA, so it's an error there. In this case, it can stabilize the work at flipping the U out a little bit, stabilize it, and then the base is removed. That site where the damaged base has been cut off by a DNA glycosylase, that abasic site, the glycosylase stays there as sort of a marker for where repair work needs to be done. And it's a marker that then recruits in the endonuclease to nick it at that spot. It will cut at the 5' side of the sugar and then cut the sugar off. And that leaves a free 3' hydroxyl group, which is what's needed for DNA polymerase to add on a base then. And so since there's now a free 3' hydroxyl, the DNA polymerase can add in the base, and then ligase would come in and seal that bit. We've already mentioned flap endonuclease, and if necessary, it will do its job before ligase. Okay. So that's base excision repair. Now we're going to talk about nucleotide excision repair, which is a little bit different. It might not sound that different, but the base excision repair takes off a base and then copes with the damage that's created. Nucleotide excision repair is going to do a little bit more than just take off a base to get things started. Nucleotide excision repair is often what's evoked by UV damage or by oxidative damage. So here you've got damage of some kind to the chain, and in this case, the DNA will actually be unwound a bit by a helicase. Endonuclease will cleave off a strip of it. So there's an actual section of single-stranded DNA flanking both sides of where that bad damage was. And then polymerase and ligase will come in and do their usual job. You know, they've got a free 3' end, you fill in and ligate and fill it up. Some of the enzymes that are needed for nucleotide excision repair were actually discovered through looking at genetic diseases. There are a number of cases where we've come to understand how our bodies work better by looking at the genetic diseases that are involved in things like that. There's a syndrome called Cockayne syndrome, and its most dramatic sign is that the children with this prematurely age, and so a child of 5 might look like they're 30 or 40. It can be very dramatic. In addition to the visible external thing of premature aging, they tend to have errors in neural development. They tend to be quite short, very sensitive to sunlight. There are 5 different places where mutations can occur and cause Cockayne syndrome. But what these genes share is that they're involved in proteins that recognize and remove stalled RNA polymerase where DNA is damaged. So you remember I said that when the damage is more than a certain size, transcription stops. That is, transcription is started along, the damage is so bad that the RNA polymerase is locked at that spot. It's stalled. And these are proteins that can recognize a stalled complex like that. You've got to get that polymerase off if you want to do nucleotide excision repair. And so if we don't have any of these 5 genes that are needed for that recognition and taking it off, the DNA doesn't get repaired properly. And these individuals have excessive levels of apoptosis because the damaged DNA is not repaired. So it's one way to think of what may be behind the normal aging process, that as errors accumulate in our bodies, that apoptosis tends to get the upper hand over the growth that's needed to keep normal replacement going. Another of the genetic syndromes that turned out to be related to nucleotide excision repair is a disease called xeroderma pigmentosum. And this is a syndrome in which people are very sensitive to sunlight. These individuals have 1,000 times greater likelihood of developing skin cancers. Not cancers for their whole body, specifically in their skin cancers. And that was one of the cues that these genes are involved in UV damage repair. One of the forms of xeroderma pigmentosa is in a DNA polymerase. DNA polymerase eta is a DNA polymerase that has a special capacity. In normal DNA polymerases, during bulk replication, you want a nice copy of DNA to copy over. Polymerase eta has the capacity to replicate through thymine dimers and to replicate across from adeoxyguanine. It's not stumped by these errors in the DNA. Partly this is because polymerase eta has a larger and more open, has a more open and flexible binding site than other DNA polymerases. It's also a little bit less accurate, is my memory. Is that correct? So its advantage is it can read through these lesions. Its disadvantage is it's not as accurate as the normal bulk polymerase would be. If you don't have polymerase eta, the cells are left unable to replicate UV-damaged DNA well, and so you end up having multiple hits of mutations, and you start to have enough mutations, you start to accumulate those necessary 4 or 5 steps that's needed to have a cancer develop, and in the skin that turns into skin cancer. I think I'm going to call it a break for there. I'll see you all Monday.